This method can help answer key questions in any research field where phosphorylation signaling is of interest, such as the cancer field, to assess changes in signaling networks after therapeutic resistance.
The main advantage of this technique is that thousands of phosphopeptides can be identified in a relatively simple and inexpensive way to globally assess the phosphoproteome.
To harvest tumor tissue, weigh the tumor and in a culture test tube, add 2 milliliters of ice-cold lysis buffer for every 100 milligrams of tissue. Then, use a handheld or benchtop homogenizer to homogenize the lysate. To reduce and alkylate the sample, heat the tube at 95 degrees Celsius for 5 minutes, then cool the sample on ice for 15 minutes. While still on ice, sonicate the lysate 3 times. Then, heat the sample at 95 degrees Celsius for 5 minutes. Place the sonication tube in a swinging bucket rotor and centrifuge the lysate at 3,500 G and 15 degrees Celsius for 15 minutes. Then, collect the supernatant and discard the pellet. To digest the lysate, use 100 millimolar tris to dilute the sample 12-fold to reduce the amount of guanidinium. For 5 milligrams of protein, add 10 micrograms of lysyl endopeptidase, or Lys-C, and incubate the sample at room temperature for 5 to 6 hours. Prepare 1 milligram per milliliter of TPCK-treated trypsin.